So the next two speakers are from a pharmaceutical industry, and the next speaker is senior scientist Adam Hyman. So Adam received his PhD from the University of Leeds under the supervision of Professor David Beach in 2018, where he was investigating the modulation of piezo-1 by lipids. Following this, Adam moved into industry, working at Charles River Labs, and in 2022, he moved to GSK, where he has been developing assays for electrogenic transporters and ion channels for hit qualification and mechanism of action studies. So over to you, Adam. Thank you very much.
Hi, can you hear me okay?
Yes.
Okay. Let me start sharing my screen. Okay, is that all coming through?
Yeah, looks good. Good.
Okay. So today, thank you for the introduction, Celia. Today I'm going to be talking about high throughput screening for electrogenic SLC transporters. Unfortunately, I won't be sharing much original data, but we'll be talking about some nanion technologies within this. So why are we investigating SLC transporters? There are many SLC transporters with a vast array of different functions and transporting different substrates, but they're a relatively underexplored target for drug intervention. And there's potential family members that have genetic links to currently for diseases with unmet therapeutic need. Additionally, some SLC transporters are recommended to be investigated by the FDA and EMA, such as OCT2, OAT1, and PEPT1, mostly due to the transport themselves of drugs. So we don't want to be inhibiting these targets to and avoiding any potential drug-drug interactions. So the first step to really finding a new therapeutic is to generate new chemical equity against your target. And so this talk is going to be focused on high throughput screening. So there's many methods of finding new chemical equity, whether it's knowledge-based and basing chemistry off previously discovered drugs. And there's many other sort of encoded library techniques and all sorts nowadays. But I'm going to be talking about the fairly old-fashioned, unbiased screening of lots of compounds. So the early discovery process for HTS, so initially we'll start with target identification and validation, ensure that our target of interest is relevant for the disease, and then we'll progress into hit identification and hit validation. After this, you'll have hits, which then go to hit to lead generation, lead optimization. So it's a little bit like sifting for gold. You start with lots and lots of material and hopefully end up with some lovely shiny gold bullion at the end. So how can we achieve this? So the main screening cascades start with tens of thousands of compounds or to millions of compounds. So for example, recent screens here have been 2 million compound screening deck. So require a primary screen capable of screening at this throughput within a reasonable time frame. So the initial primary screen will have a single concentration, single repeat of each compound. Any hits from here will then go through to hit confirmation stage in the same assay, where it will be repeated another 2 times. So we'll have an N of 3 for the hit compounds to see whether the hits confirm. Usually there's a fair amount of attrition between the primary assay and hit confirmation phases. Any hits that are confirmed will then go into potency determination, and usually in the primary assay, and may even include selectivity assays at this time. From there on, that's when the medicinal chemists start to put their interpretation of the chemical matter on things. So we start triaging compounds based on either compound purity from the screening deck, and chemistry filters such as interference compounds, frequent hitters, and whether the size and lipophilicity of compounds is acceptable for them to progress. Once we have hits from this screen, that isn't it. It's not necessarily say that these are hits. We then want to confirm these hits and qualify these hits in orthogonal assays. So we want to have an alternative technology to the primary assay to say there is a similar activity of the compound on the target in a separate technology. So this is aiming at eliminating any possible false positives that have just been taken through due to the assay technology using a primary screen, and then additional selectivity assays for related targets, rodent orthologs, to ensure any hits that do go through are active in downstream in vivo models. Once you have that and you've tested, particularly testing a newly synthesized powder of the compound, you'll then have a validated hit. So the key technologies that we currently are using for screening, in my department at least, for electrogenic transporters, are the flippers, imaging, and mass spectrometry. So with the flipper, we use a flipper membrane potential dye. So this claims to be as good as electrophysiology, although I would not suggest that to any electrophysiologists. But we can measure change in membrane potential after substrate addition with any electrogenic transporters. So if they're either sodium, proton, or even pass charged substrates themselves. The imaging requires a fluorescently labeled substrate that's either specific to the transporter or relatively specific to the transporter. So a well-known substrate is this 2-MBDG, which is a fluorescent glucose analog. And mass spectrometry, we can then measure, it's more a direct measurement of substrates within a sample. The imaging in the mass spectrometry are quite complex and resource heavy in terms of generating samples, even if they are 1536 well compatible. So this makes them quite slow, expensive, or slower and more expensive compared to the flipper. So generally our focus is on flipper assays for at least the primary assay. When developing assays for the flipper, we look key things to look out for are reagents. So we want cell lines that are appropriate for the assay, and how we express the target is also incredibly important. So generally we use either stable cell lines, or quite often we'll use BACMAMs or baculovirus transient transfections. Additionally, the substrates used, if you have multiple different substrates for a transporter, making sure which is disease relevant and also which gives the best signal in your assay. And there's also modification, sort of the membrane potential dye, what concentration to use, and also any buffer components. So if you obviously have a sodium coupled transporter, you want at least decent amount of sodium in your buffer. So the rest is fairly straightforward after this. So you want to be able to confirm no modulators and determine DMSO tolerance in your assay. So the DMSO tolerance will be able to determine a top concentration of your screen. Generally screening from 10 millimolar stocks. If you want to screen at 100 micromolar, obviously you need a 1% DMSO tolerance, and that's generally what we aim for. As we're screening in 1536 well, generally we'll start with a 384 well assay and then miniaturize this to a 1536 assay, and this requires a lot of automation, so automated flipper workflows, aspirators and dispensers, as you cannot do this manually in this plate format. And then finally, we'll have a pilot phase where you test a small selected subset of compounds just to see your hit confirmation rate and how robust your assay is. So at the bottom, I've just got an example from a recent SLC antagonist HTS we had. So we were using a UTOS background, and these cells were frozen, thawed from frozen straight into the flipper plate with BACMAM added. There was a slight modification to the buffer used to make sure we could see our substrate addition window. And the main challenge in the project was the reported inhibitor was not active when we tested it. So we had to find a modulator ourselves. Fortunately, we had some screens from related targets where we could delve into potential hits, and we've managed to find a tool at least for the screen. And from here, we managed to miniaturize the assay and in a pilot confirmed active compounds and potential assay interferers. So here, got two examples of screens that have recently finished and we have qualified hits for. So generally in the upside down pyramid, so most of the work is being done by flippers, at least up until the XE50 stage. But we have two different approaches to our hit qualification stage between the two targets. So one target was looking at the imaging technique, and another target is using mass spec and also the surfer. So why do we invest in the surfer? I think it was actually a year today that the surfer arrived at GSK Stevenage. So it's a good anniversary to be talking. So we realized that we had a major sort of deficit in our capabilities for looking at transporters. So we have flipper, we have mass spec, but no electrophysiology techniques available at a reasonable compound testing throughput anyway. So each method that we have is susceptible, has the downfalls. So particularly flipper is susceptible to off-target interference and fluorescence interference. Imaging is just dependent on the substrates available, and so it limits the amount of what transporters we can actually run this assay, use this assay for. And the mass spec is probably our most powerful screening tool. However, it's very laborious, as mentioned before, so to generate samples. So and we don't get any kinetics of how the transporter is working. It's just an endpoint read. So we invested in the surfer just so we could get this real-time transport rates and have a label-free measurement of transport. So currently we are embedding the surfer in our screening cascades. At the moment, we currently have the surfer M1 system, hoping to get 96 SE. But we're currently qualifying hits, so generally testing exemplars from hit series in orthogonal assays just to confirm their activity and compare to the other technologies. And one of the main benefits of this system for us has really been translational assays. So we've this year we've spent a lot of time measuring transporter activity from organelles themselves. So rather than having an overexpressed plasma being on the plasma membrane, we can look at the transporter in its native environment. Additionally, we're also looking at producing proteoliposomes, and the key challenge for us here has been producing enough quantity of the membrane protein to be able to use within this assay and also for any other uses for that protein, so structural studies, etc. So these are things that we're currently working on, and I'm hoping next year I'll be able to share a story with you where it includes a lot of this data. But currently this is where we're at with the surfer, and we're definitely expanding our capabilities on the system. So just want to say thank you for listening. Thank you to Nanyan for inviting me to talk, and thank you to my colleagues for helping support work on the surfer this year and the transporter projects. Any questions?
Thank you very much, Adam. And happy anniversary then, also to us, I'd say. There's a question here. I'll hand over the microphone.
Thanks for the talk. So I'm looking for the surfer currents next year. So, but one question. So you were referring to that EcoMS is labor intensive. So what's the reason for that?
It's mostly the generation of the lysates beforehand. So it's to get a decent signal, you need a lot of cells to produce to see any signal at all. So miniaturizing this is incredibly challenging, and the time to generate the we generally find longer incubation times produce a more robust assay. So it's more time, so that's why the flipper is preferred as a higher throughput screening method compared to the EcoMS.
Okay, thanks.
Thank you very much, Adam. I think we have to move on to the next speaker, and the last one in this session. We're